All right, so as Jordan said, I'm at Virginia Tech. I'm in a new biomedical research institute, the VTCRI, and we work at the interface between medical applications and material science. And today I'm going to be talking about how we're using the protochip system and integrating that also with our cryo-EM to try and understand high-resolution structures and pairing that with dynamics that we're inferring from our Poseidon work. So in general today I'm going to cover these three topics during my talk. How we're developing new tools to view macromolecules in real time, and then we're developing a new branch in liquid cell imaging with the Poseidon usage that we call liquid cell electron tomography. Then I'm going to talk just a bit about the cryo-EM work I do on the BRCA1 gene related to breast cancer, and that'll tie everything together. So as we know, macromolecules that are used in biology in particular span the gamut. People use polymers to try and understand everything in applications from fuel cells to drug delivery systems all the way to vaccines. And one thing that these have in common is that we want to understand and characterize items at these really small scales that have these vast applications in human health and in industry. And one way we do that is using high resolution imaging, in particular TEM. And in my laboratory, we started using cryo-EM. That's what I was trained in. And so what cryo-EM does in the biological sciences, they say it allows us to view small things in a big way. Everything from sections of cells demonstrated here in this tomographic slice of a eukaryotic cell. You can go through and segment out and see the different organelles in different spacings, as well as the small subunits that compose all the things that drive these cells toward life processes. So we look at whole cells, sections of cells, all the way to the molecular adversaries such as this picture of influenza virus that can infect eukaryotic cells. So we want to understand how these systems are integrated together, how pathogens can infect host cells, and then interactions that go on within the host cells, as well as things related to cancer. Using cryo-EM to do that gives us a really elegant way to look at these very small items. And so the first part of my talk and the part that we developed in our laboratory is I'm trained in single particle cryo-EM. And this is just a schematic to explain single particle cryo-EM here. We place macromolecules of interest onto a substrate, and these macromolecules might lie in different angular dimensions or in different ways in which they fall upon the substrate. And then we freeze those and we take images of those inside the electron microscope. And so what we end up with, as everyone knows, are these projection images of these different molecules in different orientations. We then collect enough of these projection images and using different computer programs and algorithms such as angular reconstitution and Bayesian statistics, we recombine these different angular projections into a three-dimensional structure. And thus we're able to reconstruct things from two-dimensional images into 3D. So this algorithm is well established in the single particle cryo-EM field. But what's not so progressive or changed a lot, there's been a lot of progress in computational methods. There's been a lot of progress in refining the electron beam, correcting the beam, adding all kinds of fun bells and whistles to the electron microscope direct detectors. But we're still using the same substrates we'd always used since the 1950s, since people started doing TEM. Carbon grids, or carbon grids with holes integrated into them. And I started my lab back in 2011, and I initiated a collaboration with Protochips, and we were using their system for in situ and doing lots of different things with in situ imaging. So we said, can we also use their chips for cryo-EM? So we started using their chips for cryo-EM, and we have this laid out here. This is a 96 well plate that we use in biology where you can have different patient samples here, and then you can just elegantly pick up the chips that you use in the in situ device and get different patient samples and then plunge freeze these and start to look at different macromolecules in doing so. The chips themselves freeze very similar to a Holy carbon grid, and so we think that imaging on the silicon nitride chips is going to give us better contrast than imaging on just the Holy carbon substrates because you have a continuous substrate. I'm going to go into that a bit further, but you can see that the chips that we use for in situ fit quite easily into the specimen holder, the side entry holder for cryo-EM. So now we think we have a more versatile substrate that's flat, nicely engineered, and a continuous surface rather than a Holy carbon grid. And so this is just us loading the chips into our specimen holder for cryo-EM. These are just all the tools that we use for cryo-EM. We keep everything under liquid nitrogen temperatures, obviously. We've frozen our substrates onto the microchips. Here we're removing this one, and then we're placing our new sample into the chip, or on the chip, into the holder. It fits quite nicely. You can then take the tools that come with the standard 626 Ganett holder, seal things up, and place them inside the cryo-EM to collect your single particle data. The chips also fit into the holder of the Titan Creo. So it's just we were doing in situ on side entry, so we're also doing here cryo-EM in the side entry holder. And what we got was quite interesting. Using the same virus specimen that we see on C-flats. So this is a typical holy carbon substrate. There are a couple different brands. We use the C-flats sold also by Protochips. But what you can see here, these little balls, these are viruses in space over the holes, as well as on the carbon substrate. So in cryo-EM, we aim to get viruses over the holes because you can see the contrast between the particle and the frozen hydrated background here is much greater than that over carbon. But when you look at how the particles look over silicon nitride, you can see the particles with much higher amplitude contrast, as well as some things in the background here. So these bits in the background, this is RNA related to the virus. So what we've done here is we've added chemicals to these viruses. So they start to do a biological activity, and they produce RNA in solution while we're freezing them. And you can actually see the single strands of RNA here in the background, and that's one of the most fragile units in biology. All the information of that is missing here in this image. And we believe that there's just a lot of Brownian motion when we're imaging here. The particles in space over holes compared to that of a continuous substrate. And silicon nitride is a really nice flat continuous substrate. It freezes really easily in the electron microscope. And just a reconstruction from one image gives us very reproducible results between that of the holy carbon film and that of the silicon nitride. And in fact, when you scroll through these reconstructions, you have a lot more detail present from the activity of the virus particles here in cryo-EM. And so what we're doing now when we prepare these cryo-EM specimen is that we're plunge freezing these biological machines into liquid ethane. And that allows us to collect images of frozen mechanisms. And that's really great. You'll get images and then you'll reconstruct these and try and piece together the live processes by which these sequence of events occurred. So it would be great if we could do liquid cell EM, which Protochips specialized in, and this is what we were working on for several years now. That allows us to maintain biological machines in liquid, and thus we can collect images of dynamic mechanisms. So it's great to have the static images because you can push these to higher resolution. Things aren't moving around as much, and you can actually get higher spatial resolution. But it would also be great to get good temporal resolution and actually see these things in action. So that was one of our goals when we first started doing liquid cell EM with Protochips. So we like to say that liquid cell EM is a new hope. Instead of looking at an item that's frozen in time and in space, you can actually see this same item in action doing different things here, right? But then someone pointed out, well, you're just showing me a snapshot of something, Deb, here versus a snapshot here. That's really not something that's moving. So I got a better example recently of my dog in Virginia. Here we can see Annie, frozen in time and space. And here we can see her being an agility expert. So if you just only had that static image of her, you could see how cute she is, but you couldn't see how great she is at agility here, where she just is like a star athlete. You know, it is Olympic season. But nonetheless, it's very different from seeing a snapshot to actually seeing something actually in motion, right? So what we want to do is observe both of these. We want to see every detail that we can at high resolution for drug design, for vaccine design, and for other properties, but we also want to see how things work, basically on the fundamental level. So now let's go into a bit of liquid imaging and how we're trying to look at viruses and such in liquid. So there were very beautiful movies shown by Jordan this morning of how this is assembled. So this was one of our very early on papers in which she showed the base chip of the Poseidon holder. You add your liquid specimen to it, then you add the top chip over top. It's hermetically sealed by these nice screws. You can start to look at things in liquid. So being a very conservative scientist at the beginning of my career, I was like, well, we need to look at nanoparticles or something like that before we just dive into looking at viruses, just to ensure we understand the beam properties, what's going on with damage, and how things move in general. So Madeline brought us these gold nanorods that we started working with several years ago. And what we could see is these gold nanorods very clearly in the liquid solution now that's sealed up inside my TEM. And this arrow is pointing to this one particle. You can see if you just look here, the beam is actually spreading the particles around because it's actually moving the liquid in which the particles are contained. And then after several seconds, things can diffuse back. If you just focus on this particle, you see this region is different from this region. So some things have moved in space. If we just focus on one particle at very high resolution, actually zoom in quickly while it's staying in place, we can see diffraction out to 2 angstrom. Here in the Fourier pattern, we have diffraction information out to 1/2 angstrom. So it tells us these nanorods sandwiched between 2 silica chips and liquid, and here I think we're using micron-sized spacers, we can still get atomic resolution. So for biology samples that are more fragile and don't have such high resolution information out to 2 angstrom, we should be able to get some mechanistic information. So we wanted to see these things move, because that's the point of doing this sort of dynamic imaging, right? So here is the first movie. I still love this movie. This is the first one we recorded where you can see these gold nanorods moving in space, and liquid, this is liquid carrying these particles in, and they start to accumulate as these nanoscale tidal waves sort of push everything together, as if you're standing at the beach and then the water rushes in, you're left with all the kelp at your legs and such. So the beam is moving the solution around. These gold nanoparticles are being concentrated. Well, these gold nanoparticles are therapeutic reagents that can be injected inside of tumors. You can use infrared radiation to sort of collectively push them around and then irradiate them quite nicely so you have a stronger kill rate of the tumor cells. So we thought, let's just do a fun experiment in which we put a cancer cell inside the TEM and then give it these gold nanorods and see what happens. So these are glioma stem cells. I have a colleague that works on glioblastoma in the lab next to me. And so we took some of these glioblastoma stem cells, we placed them upon the small chip with the Poseidon holder, and we added these gold nanorods to it, and we just started recording over time. So these are with larger spacers. The cells are a bit squished. They're not very large because they're stem cells, so they're small in nature. They are squished down in between the 2 chips. But nonetheless, we could actually see gold nanorods going in the edge of the cells themselves. So this is the edge of the cell placed inside of the Poseidon cell. Here's a mitochondria, and we can see gold nanorods have already gone inside by the time we started recording. And over a 30-second time frame, just imaging at the edge of the cell, we could see changes here in the electron scattering properties and the density that represents, you can see it better in these contour plots, changes in the accumulation of these gold nanorods inside the cells. So let's show just a bit of a movie of that here. So this was at a slow recording rate of just a few frames per second, but this thing happened over the course of like several minutes. So it's a little more disjointed than I would want it to be. But we can see in different areas in space that things are starting to accumulate as the water is sort of moving around, and we're imaging through the entire thickness of the edge of the cell here. And what this plot showed is that we could quantitatively go in and see these particles moving around. So we know that we could probably observe nanorods entering the cell, and we could also see areas in which they're exiting the cell, because these cancer stem cells are actually known to extrude drugs. That's how they become therapeutically resistant. So we can see more nanorods moving around inside of the cells. And then people always ask me, well, you know, why don't they move more? It's because the cell is full of stuff that you actually don't see, because you can only see the nanorods here with the high contrast. So it's actually packed full of stuff, so you don't expect to see a lot of things moving around. But nonetheless, we could see things coming in the paper that we published, as well as things going out at the edge of the cell. But if we want to understand host-pathogen interactions, getting back to the viruses now, we needed something where things didn't flow around very heavily. And so we worked with protochips as they were designing this next generation of chips. And so this next generation of chips, as well as this next generation of holder, this is the fancier movies that Jordan showed this morning, is a single O-ring system, a base chip, a top chip. This part isn't much different. It's the O-rings now that are much more simplified in the system that make life much easier for users. So you have the single O-ring design, the base chip, your liquid sample, and then the top chip. It's sealed up and placed inside the TEM. So one thing that we really like to use now for these small virus particles compared to larger cells are these microchips, these base chips with integrated microwells. And so these microwells, it's kind of a bit like an ice cube tray, and then the solution goes in and fills up the tray at different levels. And so the spacer that we used here was only 150 nanometers thick, and these microwells are 10 microns in X and Y. And so you have very thin Z dimension, but you have a large X and Y dimension. So things should be more sealed off than they were with these large micron size distances. So here we can see, what I'm going to show are magnetic particles that are used as contrast reagents in MRI. But let me just point out that this cell is mostly liquid. You see a little bit of dryness. This has a lot of dryness. You see just a bit of liquid. And then this has a thick gradient of liquid across it. So we can mostly fill these cells up, or you look for ones that are more full or the right thickness of liquid. And so these reagents are magnetic now, and they're kind of jumping around, but they're starting to kind of coalesce toward each other. You can kind of see they're kind of coalescing here together because they're magnetic. So the movement of these particles is a lot less severe than that of the gold nanorods using these more confined microchips. But we still wanted to hold like virus particles in place and then watch them do their job. So we tethered them now using molecular protein adapters and antibodies. So antibodies are natural protein reagents that are specific for the coding of our viruses that we're looking at. So we added a different coding system with antibodies against our virus, and we could see in doing so, we were able to tether them and concentrate them quite strongly. These are our viruses. This is just a negative stain image to ensure the antibodies are working now. And so with these antibodies, they're called IgGs, we can tether virus particles specifically to the chip, and without them we get little to no virus binding. So we have our specificity now, and we have a way of tethering them, sort of holding them in place onto the chip. So let's look at these guys in liquid now. So here we go. We're gonna look at these. This is our first snapshot that we ever took of viruses in liquid. We quickly did a 3D reconstruction, and we got a couple of different classes on one single image just to make sure the integrity of the particles were in place. From there, we knew if we had, these very clear particles with high contrast, similar to what we saw in cryo-EM, and good 3D reconstructions on individual images, that we would have high quality data coming out of that. So let's see here. So we have our viruses tethered into our microwells. We take a series of images, all in the same viewing area, just image, 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 like you would in any other process. 4 to 10 images per second. And over the time spent in 10 seconds, we had really good data. And what we see attached to these virus particles are, sometimes we see these strings attached, and that's the RNA, indicative of viral activity attached to the particles. And some had them and some didn't have them. This one has RNA, this one doesn't have RNA. And so we could color these in different colors. And although your eyes can't necessarily distinguish major differences over the time course, if you take and focus on one of these and then look at the internal scattering properties, we can see a lot of information, in fact. So let's do that now. Let's focus on this one particle that we know has RNA attached to it. What we're going to do is a density threshold where we just look at the highest electron scattering properties. And that's indicative of the viral genome. So this is all RNA inside the virus, and we can see the RNA moving inside the virus now. And we still see it attached here, so we know the beam dosage isn't so high that we're destroying single strands of RNA. So we're able to actually go through and quantitatively map how the internal components related to the genome of this virus is changing. So quantifying that now, what we could see on different representative particles that we chose here. Here's the density threshold, and here's a contour map of that. We could go through and calculate using heat mapping the mobility of each of these units, meaning how did the pixels displace next to each other in time and in space. And then I made my students go through a series of quantitative analysis because it was fun for them to write different MATLAB codes to do this. So what we could do is now map these in a more quantitative fashion. And just the main point of this is that the more RNA that we saw attached to the virus particles, the more genomic material extruding out of the virus particles, the greater were the pixel displacements in the heat mapping, meaning the more the virus particle was moving, the more RNA was being extruded out of it. So it's indicative that we're actually capturing images of a biological process. So we're very happy with that. We can get down to the molecular level and watch a genome move inside a virus particle. So that ended our proof of concept of what we're doing there, and now we're pushing this to a higher level, which we want to take and produce real-time movies at higher frame rates, actually see more intricacies of these virus particles and things being extruded from them. We want to produce movies now of viruses actually entering host cells, similar to how we are producing movies of nanoparticles entering host cell. Viruses are one of nature's nanoparticles. So let's watch those enter host cells now. And then we want to do experiments in which we produce this reaction with the virus particles. They produce this RNA, and then we want to capture that RNA with another assembly that will turn that RNA into protein, and those are called ribosomes. So we have a lot of different biological projects in the way in which we want to move this forward. But you might say, and this was also one of my concerns, on the images in which you're going to watch viruses go inside of cells, how do you know they're actually going inside cells? They're not just landing on top or below the cell, and you want to put everything together in 3D, right? You want to see something go in, and not just the top of it, or you're confused because it's like above it and you don't really, you know. So the way to do that is you have to tilt the sample in the electron microscope. So that brings me to the next topic, in which we're developing liquid cell electron tomography. So electron tomography is a rather older concept in cryo-EM, and this is how it's performed in cryo-EM. People have their sample that they want to observe. They place it onto an EM grid. They plunge freeze it, similar to the processes I was talking about earlier. And then you take the frozen grid and you place inside the electron microscope. And instead of just collecting images in this single plane, in which you use the angles to reconstitute it as a single particle EM, you tilt your specimen inside the electron microscope. So then you would tilt from 70 through 0 to minus 70 degrees. And you end up with this image stack. So within this image stack is the information as you're tilting through the z-axis. You could take and then reconstitute that using programs established in the cryo-EM field and develop three-dimensional renderings of your specimen. And that's electron tomography. And here's a great example from Grant Jensen's lab, in which he's imaging bacteria, and you can see clearly the different membranes surrounding the bacteria, the flagella, and then using a lot of different type of averaging techniques, he was able to go through and segment out what different items were that constituted the components inside of this bacteria. And so that's what we want to get to, but we want to see these things in 3D as things are entering and taking over these host cells, or as bacteria are being infected or human cells are being infected. So we're developing this technique now for liquid cell imaging to do using the same principles. So what we do is we have an EM grid or a chip placed on the base of the holder. We add a liquid sample and then we put a grid over top of it. So it's a unique configuration in which you have a chip, liquid, and a grid, and then that's sealed together and placed inside the electron microscope. So we use a grid instead of a second chip now because we get more of a thin layer in the z dimension. You can actually tilt easier and get higher tilt information and reconstitute your tomogram at slightly higher of a tilt. So we can go up to 60 degrees. Our holder can go to 68 degrees. We can do 60 with high reliability. And we can just tilt a little bit more by replacing one of the chips with a grid. So the sample we started as a model system to play around and look with as part of a collaboration with my colleague in biology is bacteriophage. And bacteriophage are viruses that infect bacteria. And so the system she's interested in is this really cool phage where here's the head of the phage and some fibers attached to the head. It has this really unique looking tail as well as tail fibers. And so the tail fibers help it attach to flagella, and then that brings it along and somehow the phage joins up with the bacteria cell and can inject its DNA through its long tail into the bacteria and take over the host cell. Or at least that's the theory, but no one's actually really seen that. So we said, okay, these are really unique looking items. Let's just look at these in general in liquid and then try and look at them trying to infect a bacteria cell. So this is our first tomogram we did of bacteriophage in liquid. So this is some of the buffer solution. We're tilting here so you can actually see it being tilted inside the solution, but you can clearly see this little key-shaped item is a bacteriophage. We can go through and then segment that out. Let me go to the next one, the next movie. So we identified where it was in the tomogram. We can go through and then segment it out and remove the rest of the background, and then we'll just see this item rotating in space. So it's kind of dark here, but we've segmented out the bacteriophage. It's just rotating in and out of the plane from the tomogram. So we've got the right algorithm and the right experimental setup to see things in liquid and in 3D. So here's the bacteria that they naturally infect. And so we put bacteria onto our chip. We added bacteriophage and we said, what's going on? What can we see? This is actually the flagella of the bacteriophage. A lot of them are hopping onto the flagella and interacting with the flagella, and then somehow it's thought that they interact with the flagella, probably because it's more floating around in space, and then they whip around and enter the cell, or not enter the cell, attach to the cell and inject their DNA. It's not known whether they attach to the cell via their tail or their head. Different phages use different modes of operation. And we still can't tell, but we can just see these sort of heads attached to these little convolutions upon the bacteria. So let's tilt it and see if we can't get some better insights from that. So here is our first tomogram where we actually tilted the whole system. You can actually see more details coming into view where you're tilting these. And these are surrounded by liquid as well. Some areas are thicker and some areas are thinner. We could go through and just map out and identify where these different areas are. Here's phage, here's the individual bacterium, obviously. Some are coming in to infect, and it looks like some are attached or some are above or below the imaging plane. These look like they're attached somehow to me. And they also look like they're attached in the higher tilt angle. Here's just a representative image at 45 degrees. We can go through and also do the same electron density filtering that we did for the virus particles I showed earlier, but these are in a slightly different color range, where yellow is high density, all the way to white, which is low density. So white is the solution here, and this is slightly thicker solution. So you can see the slightly thicker solution surrounds the bacteria. And then the higher density particles are what we believe to be the genomic material inside of the bacterium itself, as well as the genomic material that's packed inside these little phages as they're attaching here. So you can clearly see that they're attaching, and it looks like their heads are really attaching without the tails. But this was our first tomogram of this. We've gotten much better right now in time, but I still like showing this one because you can kind of see these would make you think they're attached, but they're actually below the plane. And these are really heavily attached, right at the central plane where we were looking. These are off in space as well. You don't see that total phage. This is just a central section of the tomogram. You still don't see the whole round phage like we would see if I was showing you a 3D here. But through the central section, you can actually see the attachments, which is why I showed the central section in this movie. And here's the central section. So these are pinpoints of those phages, those little round heads. And these are attached. Here they're starting to attach, but these are clearly below the plane. So we now have a way in which we can, with certainty, see how things are attaching by tilting in liquid within the electron microscope. So we want to understand mechanistic insights of these host pathogen interactions. Is it the tail, or is it the head that attaches? And how does the DNA from the phage get into the bacteria? And we also think that this would be really good to go back and do some more work on therapeutic delivery to cancer cells, now that we have the tomography and everything in place. Now, it might be that the time regimes are so fast at these things that we miss some important information while we're tilting. But you could do tomograms now with just a few minutes. It doesn't have to be like several hours of tilting. So we're working to do rapid tomography as well, so we don't miss all of these insights that we want to observe. So here's some things that are a work in progress for the lab. So going now, I'm going to just transition and talk briefly about some of our more heavier cryo-EM work to understand the breast cancer susceptibility gene in cancer. And so this is just a little bio for the engineers and materials people in the audience. Just be entertained by the pictures. It's cool. You don't have to go deeply into the mechanisms. But I would just like to show that we can look at these smaller molecules now using chips for cryo-EM as well. I showed that we can use the chips for cryo-EM of these large viruses that are highly symmetric. But now we're using the chips for cryo-EM of these very small 300 kilodalton proteins, and we're getting quite good results with them. So I just want to show that briefly and why we're interested in this. So here's just a very rudimentary overview of human cells. within the cells, the command center that controls that contains all of our DNA and controls how organs and tissues are laid out is in our genetic code. That's defined by DNA, which is wound up around proteins and nucleosomes within chromosomes. And so certain pieces of DNA have to be revealed and not revealed in order for genes to be activated on a daily basis for every process in our body. And through some of these processes, DNA can get repaired and from damage on a daily basis. So when DNA is damaged, you better hope that your repair mechanisms are at least equal to the rate of your DNA damage that occurs. So in every cell of your body, you get 500,000 bits of DNA damage on a daily basis. That's a lot of repair that has to occur. And the rate of repair has to be equal to that of damage for cells to be maintained in a healthy manner. So when the rate of repair is not equal to the rate of damage, eventually the system wears down for troubleshooting and keeping things in check, and DNA damage overtakes a cell, and you end up with disease formation. So this might take a series of years, it might take a series of weeks, but fundamentally you have to maintain your genome in a very stable manner. So it's a really delicate balance inside the nucleus of the cells, maintaining healthy DNA and warding off adversaries that want to destroy it. So this is a constant struggle that we call placed between oncogenes and what's called tumor suppressors. So oncogenes are things that help promote cancer that are ingrained inside your DNA. Either proteins are turned on to be expressed too high or genes are expressed too high in a manner in which they're not regularly controlled. And it's the job of tumor suppressors to actually control that and keep the oncogenes in balance. And some things can easily throw off this balance between tumor suppressors and oncogenes. And those can be a series of both internal or external factors. And so some of the factors that particularly influence breast cancer that we're interested in are the combination of genetics. If somebody inherits a family gene or something that makes you more disposed to cancer, the inappropriate breakdown of hormones creating free radicals, the level of which we get DNA damage on a daily basis, or stress. If there's any graduate student in the audience, you know a lot about stress, I'm sure. Sadness, depression, pain, all of these things that go along with graduate school or being a postdoc. So it's just when you have a combination of all these stress factors at the molecular, cellular, or organism level, as well as inappropriate breakdown of hormones creating free radicals that can damage DNA and genetic predisposition, this is a really trifecta to enable disease processes and throw off that ratio between tumor suppressors and oncogenes. And the tumor suppressor we're really interested in is called the breast cancer susceptibility protein. So when you have mutations in this protein that are inherited, you're 50 to 60 more times likely to develop cancer throughout your lifespan. So 50 to 60%, that kind of just sounds a little bit more than a flip of a coin, right? So there's got to be something about it that's pushing it in one direction or another. It's usually these items. So what was remarkable to me, I started reading about the BRCA1 gene. It was really popular right when I started my lab. I started doing a bit of reading about it. And there's a lot known about the BRCA1 gene and mutations of it causing cancer, but the gene codes for a protein, and there wasn't much known about what the protein's actually doing down at the molecular level. So that sounds like a really important structural biology problem. I was lucky enough to convince NIH to give me one to do cryo-EM on it, because nobody had looked at the structure of this very popular cancer protein ever through through structural biology. I could not believe it. It's like I thought, oh, that seems like a really important thing to look at, because this gene, when it's mutated, not only leads to primary cancers, but metastatic disease, and this is what really kills people, is metastatic disease. So we want to understand what goes wrong when cancer develops and then how it spreads throughout the body. If you have these mutations, you might be 50% more likely to get it, but when cancer recurs, and in less than 3 years, these are the number of survivors in less than 3 years out of 10. So if it comes back in less than 3 years, and it always does with BRCA1, your survival is very minimal. So to understand the protein, we thought, let's isolate it and look at it. So the BRCA1 protein has this series of amino acids with these different domains, and it binds exclusively with its binding partner named BARD1 in the nucleus of cells, and they function in tumor suppression. And mutations in BRCA1 sort of attenuate that function, and you don't get full tumor suppression function. And that's one of the mechanisms in which things go wrong. So we thought, let's look at this and see if we can get some insights as to why this mutated protein can cause these problems. So we grow cancer cells, we isolate the protein from the nucleus of cancer cells, shown here in this red dot. Then we look at them in the electron microscope, we do cryo-EM, and then we reconstruct them in 3D. And our first low-resolution structure was published just late last year, as shown in this cartoon here. So the EM density map is this squiggly looking blue thing. But when I rotate it around, let's see, I think that's a movie. Here's a molecular envelope. This is only a 15 angstrom map, but we're able to discern the N-terminal domain from the C-terminal domain, where all the mutations are in the proteins that lead to cancer. And actually go through, these were determined from cancer cells from patients. And this is the same reconstruction of cancer cells from patients with a mutation. So what this mutation does, this is a very popular mutation. It cuts off the last 10% of the protein here. It's truncated, and then that affects the function inside cells. So looking at the structure of the mutated protein, this is just also showing where the mutation is in the protein with the red star here. This biochemically says that we produce the protein, it's pure, and it interacts with its binding partner. Here we can see the particles in the electron microscope and class averages and projections of our 3D structure. So what was interesting about the protein, it looks very similar. It has this clamp-like shape. It looks similar to the wild-type protein, except for this small bit here is truncated, as I said, and that has implications in the function of it, and that it can't perform its job properly. But also what we learned about that protein was that we could restore the problems in which it doesn't do its job properly. Wait, I think this slide is out of order. So, okay, here we go. So under stressful conditions, remember I said genetics, hormones, and stress. Under stressful conditions, the mutated protein gets this lump added to it here. So this lump is actually a signal that causes the mutated protein to be degraded inside the cell. So by this mutated protein being degraded, it can no longer function as a tumor suppressor. So what we did was we biochemically removed this tag from it using an enzyme, and then the protein looks normal again. So this is the difference, this yellow here, between the unhealthy protein and the restored protein. So biochemically, we were able to remove this, we call it a hotspot area, this tag for destruction of the protein. And it goes back to its normal structure. And we've recently just reported that its structure looks normal and its function looks normal. So we can restore this mutated cancer protein now in cells. And we hope that this is a new type of preventional therapy that we could use in the future. Still got a long way to go, but I think it's very interesting that we're the first to show that let's look at it, there's something wrong about the way it looks, let's fix it, and then it restores back to normal. I thought that's a very great way in which the structure can inform new hypotheses. We tweaked our biochemistry prep, and then we're able to isolate also from native cancer cells their first active p53 complex. That's also a tumor suppressor. I guess my time's up. This is my last slide. So this is the first active p53 protein that we're able to isolate from cancer cells as well. This is also a very small molecular weight complex, 250 kilodalton, and it's asymmetric, but you can see the averages look quite nice. And this is a low resolution map of our first reconstruction of it. What we see here in blue is a broken DNA strand in which the p53 is bound to. This is just a fragment of the structure, but we can see p53 binding to native DNA while it's actually fractured inside of cells. And we just have a little movie going through the cross-section of that map. There's extra density in the map that's not accounted for in the structure that's interpreted here, because the X-ray structure doesn't have all the bits and pieces of it as the full-length structure has. And we're just going to scroll through this reconstruction as well. So what we're able to do is look at these cancer proteins in new ways directly from cancer cells using these new materials provided by protochips. So what we're eventually going toward is understanding how other mutations impact BRCA1 for repair, testing these responses in cells, and see if we can get some insights as to how we could help things with metastasis. So what we're totally developing, I believe, is a structural oncology toolkit that allows us to go from cells, extract the nuclear material, and then directly look at macromolecules as they would natively appear. And in doing so, what we're trying to develop is now a structural oncology imaging center that we call SONIC, which we want to unlock the secrets to cancer proteins and look at things in a whole new light. So I'll stop here, and I said that was my last slide, it's my last data slide. But here's our institute. My lab is on the second floor, and we're lucky to be funded by these nice organizations. And here's a picture of my research team. So I'm happy to answer any questions that you might have about the Poseidon system, the use of chips for cryo-EM, or our newly up-and-coming tomography that we're working on. All right, and thanks very much.